Dear students, आज हम पढ़ा what is plasmid. आम समस्त जानी ऑलरेडी अदर वीडियो गुड़ा शेयर करी रिगार्डिंग माइक्रोऑर्गेनिजम्स. So plasmid कौन थाए और तार different functions, तार types. So let me start the topic plasmid. So plasmid is a small circular piece of DNA that is different than the chromosomal DNA, which is all the genetic material found in organisms' chromosome, and it replicates independently of chromosomal DNA. Plasmids are mainly found in bacteria, but they can also be found in archaea and multicellular organisms also. And plasmids usually carry at least one gene, and many of the genes that plasmid carry are beneficial to the host organism, although they are separate genes from their host, and they are considered to be independent life. So this is regarding your plasmid structure. You see, this is the bacterial DNA. This is a bacterial cell. So these are the plasmids. They are circular ring type structure. This is called plasmid. So general properties of plasmid. So plasmid are extra chromosomal, double-stranded, autonomously replicating nucleic acid molecules that are distinct from the chromosome, and they exist supercoiled. I have already seen you the photos, and that is closely circled or closed circle, you can say. Nicked open circle is also there, and also linear. So it depends on what type of bacteria you have, and accordingly the structure was like that. So they are found in both gram positive and gram negative, and individual cells can harbor one to several plasmids based on number of plasmids per chromosome per cells. That is, we can say the copy number. So there are two classes: low copy number like F and large plasmid presented 1 to 2 per cell, and high copy number generally smaller plasmid with a 40 or more copy cell. So these are the properties of the plasmids, and their size ranges from the kilobase pair to 1,100 kilobase pair, and the plasmids can carry few to hundreds of genes. They show very little or no homology to host chromosomal DNA sequence. They are not essential, and they usually do not encode essential genes. However, the plasmid gene may give the bacterium a selective advantage. So these are the different advantages and the functions of plasmid. So plasmid, what it does in a bacterial cells, so they replicate independently of chromosome and are maintained in daughter cells. The protein needed for plasmid replication may be plasmid or chromosomally encoded. The cells can be cured of plasmid by ethidium bromide. These are the chemicals by lysis procedure, or when the plasmid is isolated, this type of things were using for the process. So these are the types of plasmids: fertility plasmid, that is F plasmid for conjugation; resistant plasmid, that is called R plasmid, that is antibiotic resistant, that's why it's called the R; and pol plasmid encode colicins, that is called bacteriocin; and virulence plasmid, that is encode determinants required for the pathogens; and metabolic plasmid mediated degradation of toxic compounds. So these are the different types of plasmid. So what is epigen? Epigen is also an important part of the plasmid. Sometimes F factor plasmids get integrated into bacterial chromosome. So I have seen you the structure. Accordingly, that F factor plasmid is integrated into the bacterial chromosome. Such plasmids are called epigen, such as HFR, that is high frequency. That is high frequency of recombination. HFR, there is a full form of HFR, high frequency of recombination. So this is F factor. When it integrated on the bacterial chromosome, it changes to HFR. And the next slide, that the plasmid transmission. How the plasmid transmission to one to another. So plasmid DNA sequence can be transmitted among bacteria by transformation, transduction, and or conjugation. So these are the three important transmitting factors or process: transformation, transduction, and conjugation. Since plasmid replicate independently, recombination is not required for inheritance of trait. Because it's self-transferable, or conjugative plasmids carry genes needed to provide all functions for their transfer. So these plasmids tend to be large. Many plasmids are incapable of self-transmission but can be transferred at high frequency from cells that carry self-transferable plasmids. So in this case, we can give an example that in E. coli cases, here the transformation is not occurred, but transduction and conjugation, this type of things is happening to E. coli also. So DNA exchange occurs in nature by transfer of certain plasmids called conjugative plasmid from donor cell to recipient. And conjugative DNA transport requires cell-to-cell contacts. So the best studied conjugative plasmid is the F factor of E. coli, because E. coli is a model organism for all the experiments. That's why its lifespan is very short. So there are 3 first reasons why E. coli was chosen. And transfers of F plasmid is initiated by interaction between the sex pilus of the donor, that is F plus cells, and receptor of the recipient, that is F minus cells. The F plasmid is copied by the rolling cycle replication and delivered into the recipient. So like this, the conjugation process is. And these are the role of plasmids. One is conjugation, bacterial productions. This is one type of plasmid, PBR 322, and here these are the sites, E. coli R. These are the binding sites, E. coli R, HIND3, so BAM HI, and this is the degradation of toxic compounds, symbiosis with eukaryotes. Then antibiotic productions, resistance to antibiotics, resistance to heavy metals, introduction, that is in E. coli, hemolysins, adherence and colonization factors, serum resistance protein, catabolic and anabolic pathway, resistance to uverase by way of DNA repair enzyme coded by plasmid, then have transposons, process reservoir, bacterial. So these are the different many roles of plasmids. Why we are taking plasmid for the construction procedure. So how the plasmid should be isolated? Isolation of plasmid molecules by cesium chloride, ethidium bromide, equilibrium gradient centrifugation. Mainly this is in the centrifugation process, the plasmid should be isolated. So this is a lysis precipitation, and this is binding by solution, and finally we get the pure plasmid. So these are some applications. There are important tools in recombinant DNA technology or genetic engineering. So some are given below. So production of useful substances like hormone, enzymes, antigen for vaccines. These are the applications. And factors for gene replacement or regulation of mammalian tissue. Identification of gene sequence important to virulence properties. Identification of sequence important to gene expression and functions. Plasmid profiling is used for epidemiological typing of bacterial strain. So these all are about the application. So this is here all with plasmid details about. So ଯଦି କିଛି ପ୍ରୋବ୍ଲେମ୍ ହେଲା କିମ୍ବା କିଛି କେଉଁଠି ପ୍ରୋବ୍ଲେମ୍ ଅଛି ତ ମୋତେ କମେଣ୍ଟ ବକ୍ସରେ ଲେଖିବେ, ହେଲେ ମୁଁ ନିଶ୍ଚୟ ଆନ୍ସର୍ ଦେବାକୁ ଟ୍ରାଏ କରିବି। ଥ୍ୟାଙ୍କ ୟୁ।